Today I'll introduce the Calcium City, a 5-level framework for understanding calcium signaling and the channelopathies that disrupt it. In this image we see a neuron. Calcium ions travel its dendrites and trigger neurotransmitter release at the synapse. That single ion is our subject. Over the next few minutes we'll walk the city street by street, from the quiet resting baseline to the sirens of calcium transient, to the IP3 receptor fire station, the mitochondrial cleanup crew, and finally the exit doors. The goal is a mental model precise enough for the clinic and clear enough for family conversation. Let's start with the premise that calcium is a language, not just an ion. It regulates events on 2 very different timescales. Millisecond electrical excitability at one end and long-term gene expression and development at the other. The reason one messenger can do both is baseline. Resting cytosolic calcium is held extraordinarily low, so a brief channel opening produces an enormous relative signal against a quiet background. Cells read that signal through 4 systems you can see in this cross-section. Entry at the membrane, release from the endoplasmic reticulum, buffering by mitochondria, and extrusion back out. Our framework maps those 4 systems into 5 levels, and then asks what happens when IP3 receptor channelopathy corrupts them. So you might ask, why calcium specifically? Look at the gradient. Outside the cell sits roughly 1 to 2 millimolar calcium, inside around 100 nanomolar. That's a 10,000-fold difference maintained continuously at real metabolic cost. That gradient is stored potential. A small, brief opening of any of these channels, voltage-gated, ligand-gated, store-operated, produces a large and unambiguous change. Calcium also binds an unusually wide range of proteins, and the cell controls it precisely in both space and time. That combination is why one messenger can serve fast events like excitability and contraction, and slow ones like differentiation and development. Now a word on why the analogy earns its place. Calcium signaling is not one molecule doing one job. It's pumps, channels, receptors, buffers, and organelles acting in a precise sequence, which is exactly what a city infrastructure does. Mapping those components onto streets, sirens, fire stations, cleanup crews, and exit routes makes an invisible system visual. More importantly, it makes interdependence obvious. When IP3 receptors misfire, the trouble doesn't stay local. Mitochondrial buffering and calcium extrusion are disturbed too. For students, for families trying to understand a diagnosis, and for clinicians explaining one, this gives a shared mental model without sacrificing accuracy. Now here's the mapping made explicit, so nothing stays metaphorical. Quiet streets are the low cytosolic calcium held by pumps and buffers. Sirens are calcium transients, microdomains, waves, and oscillations, each carrying a different instruction. The fire station is the IP3 receptor on the endoplasmic reticulum, a ligand-gated channel releasing stored calcium. The cleanup crew is the mitochondrion with its uniporter, whose local uptake shapes how big and how long each pulse is. And the exit doors are the PMCA and NCX, restoring the resting set point. Every city term on the left has a real structure in the middle and a real function on the right. Keep the table in mind as we go. This is the route we'll take. 5 levels: the resting city, the sirens, the fire station, the cleanup crew, and the exit doors. The dashed line matters as much as the stops. Each level depends on the ones around it. A pump failure at level 1 changes how a transient looks at level 2. A misfiring receptor at level 3 overloads the crew at level 4. And impaired extrusion at level 5 never lets the city return to quiet. That's the central claim of the framework. Disruptions at any single point ripples through the whole system, which is why channelopathy symptoms are so multi-system rather than local. Level 1 is the resting city. Cytosolic calcium is held roughly 10,000-fold below extracellular levels and well below endoplasmic reticulum stores. In this figure, you can see how that's done. PMCA and NCX at the plasma membrane pushing calcium out, circuit pumps filling the endoplasmic reticulum store, and cytosolic buffering proteins mopping up the rest. This is active ATP-consuming quiet. And when that quiet is chronically stressed, by leaks, by pump failure, by a channelopathy, the cell doesn't simply tolerate it. It remodels, gene expression shifts, receptor density changes, organelle structure adapts. The practical consequences is that a cell with an IP3R channelopathy is operating closer to the edge of calcium balance even at rest before anything has happened. Level 2 is the sirens. Signals rise briefly and precisely, and their form is the message. A local microdomain says one thing, a propagating wave says another, a train of oscillations says a third. Amplitude, frequency, and location together encode the instruction. Mechanically, surface receptors generate IP3. IP3 binds its receptor on the endoplasmic reticulum, and calcium is released. In excitable cells, voltage-gated channels emit calcium as well. Then pumps, exchangers, and mitochondria restore the baseline, which is what makes the next signal readable. When channels misfire, the language itself is distorted. And because these same signals guide development, distorted signals alters how circuits form, not just how they respond today. Level 3 is the IP3 receptor fire station, the heart of the framework. Most signaling calcium is stored in the endoplasmic reticulum, and this is the door it comes through. You can see IP3 bound to the cytosolic regulatory domain at the top, and calcium flowing through the open channel into the cytosol below. Whether you get a discrete puff, a propagating wave, or sustained oscillations depends on IP3 concentration, local calcium, and regulatory proteins. Isoform identity and where the receptors sit determine where signals begin and how far they spread. And here's the clinically important point. Pathogenic IP3R variants disrupt endoplasmic reticulum release even when standard imaging looks entirely normal. The building is standing, the doors are wired differently. Level 4 is the cleanup crew. Mitochondria positioned near release sites take up calcium through the mitochondrial calcium uniporter. You can see that happening here at an endoplasmic reticulum contact site. They act as local sinks, and in doing so, they sculpt the signal. How large the pulse gets, how long it lasts, how fast the cytosol clears. Uptake also feeds ATP production, so this is buffering and metabolism at once. But the capacity isn't unlimited. Excessive or prolonged calcium entry stresses mitochondria and compromises cellular health. In a channelopathy, abnormal firing forces this crew to work in non-physiological rhythms, and over time, that models how the cell handles calcium across development. Level 5 is the exit doors. Two transporters do the work. PMCAs offer high affinity, fine-tuned control, and matter most in neurons where precision counts. The sodium-calcium exchangers move much larger loads quickly, which is why they dominate in heart and brain. When extrusion is impaired, calcium stays chronically elevated, neurons sit near their firing threshold, mitochondria face repeated loads, and pathways meant to fire briefly become tonically active. That tonic activity drives long-term changes in gene expression and vulnerability. Channelopathies push these systems into chronic overwork, and the result is a shifted developmental set point. Not a single bad day, but a different trajectory. To bring it all together, calcium is a precisely regulated language that coordinates moment-to-moment excitability and long-term developmental programs at the same time. Quiet streets, sirens, fire stations, cleanup crews, and exit doors map cleanly onto buffering, transients, IP3 receptors, mitochondria, and extrusion. In IP3R-related channelopathies, a disruption at any one of those levels ripples through the entire city, which is why altered signaling reshapes how cells grow, communicate, and respond over time rather than producing one isolated deficit. That's the value of the framework. It gives clinicians, students, and families a way to interpret symptoms, developmental differences, and therapeutic strategies as part of one coherent system. These are the primary sources for the framework. Berridge, Bootman, and Roderick's 2003 review covers signaling dynamics, homeostasis, and remodeling. Blaustein and Lederer 1999 remains the standard reference on sodium-calcium exchange. Clapham's 2007 cell review is the best single overview of calcium signaling. Foskin and colleagues 2007 is the definitive treatment of IP3 receptor release channels and underpins level 3. And Rizzuto and colleagues 2012 covers mitochondria as sensors and regulators of calcium signaling, which is the basis for level 4.